Hi, everybody. Phil Yeski, UMDF Science and Alliance Officer. Welcome to the May 2024 Bench to Bedside webinar. The slide you see up is very relevant. Today, we're going to be going over the scientific program at Mitomed 2024 later in June. It's an opportunity to give you a little bit of an idea of the scientific platform, and we're joined today by all of our scientific co-chairs. So we look forward to sharing that with you. But a little bit of housekeeping before we get started. I also like to remind you a really important part of our conference are the poster sessions. And we've got two really great sessions planned both Wednesday and Thursday at the end of the programming those days. We had over 100 posters submitted, really high quality research. I think the scientific chairs and myself here at UMDF, we really enjoyed going through it, and we selected a nice handful to also have oral presentations. So be sure to check that out as well. Also want to mention that last year we started a clinical research pavilion as another part of our conference programming. We're coming back this year, bigger and better. We have a lot of the same studies that are coming back, but at the same time, we've also added a few additional ones. So this is a wonderful opportunity with the patient and family community at the conference to get them engaged in clinical research. It's a way for us to advance these studies, but importantly, for these patients to learn what it's like to participate in clinical research. This all falls under the heading of trying to create a culture of clinical trials within mitochondrial disease. And so the clinical research pavilion will be a big part of that. So if you have patients, please do make them aware of that. I'm going to let Zwela talk a little bit about some of the CHOP activities taking place.
Thank you, Phil. And so this is the second time we're doing this. It was extremely successful last year where we conducted the first pavilion. There we had the gate mat, see the photo on the top, as well as these balance sensors that are a separate device. However, this year we're adding on a third device, which is foot insoles that pretty much does the same thing as a gate mat, but the attraction there is that remote monitoring can be conducted during a clinical trial. So essentially, we're wanting to collect more data, not only on patients, but also on volunteers. So please, researchers, participants, please do volunteer. It would be really wonderful. We also have new methods of measuring mitochondrial function that we're working on at CHOP, funded by the Department of Defense. There we'll be collecting breath and conducting pupillometry testing. So please, anybody that can volunteer, sign up. We would really love to see you. Thank you.
Yeah, and as we get through the programming for the week, you'll see we're providing some feedback from what we learned last year at the Clinical Research Pavilion. I think that will be a regular part of the conference as well going forward. Quickly, just want to mention our patient populated registry. It's the only worldwide patient registry for mitochondrial disease. Critical, as I talk about clinical trial readiness, that we have a patient populated registry where we have the chance to characterize them both genotypically and phenotypically and really gather the patient perspective. So please encourage any patients you may have interactions with to join. Okay, just to wrap up the introduction, some brief housekeeping. View options. We always make the suggestion to select speaker mode so that you can see the speaker and their slides. Closed captioning is available on the menu at the bottom, as well as caption translation. Encourage you to take advantage of these features. And with that, this is what we're going to cover today. Looks like a lot in an hour, but we're going to move through it relatively quickly. Our goal today is to just tell you enough to get you excited about the conference. If you haven't already registered, to perhaps convince you to register and come and spend a few days with us in Cleveland this year. We'll move through each of these platforms one by one, and then we'll wrap up at the end to see where we are. If you have any questions, we would suggest putting them in Q&A. Okay, so we're going to get started. Platform one, I'm going to turn it over to Thomas Kosic, who is going to tell us about that platform. Thomas.
Thanks, Phil. Hi, everybody. Good afternoon. So I'm Thomas Kosic, working at Mount Sinai Hospital in New York. And this session is about advances in disease models for investigating mitochondrial diseases. And why we actually propose this platform is that you may know that models, for mitochondrial disorders, they have their disadvantages and also some of the challenges, especially when we talk about mitochondrial DNA-related models. This has been really a big hurdle in the field and really hindered, progress, because it's still very difficult to model it in animals and even in maybe other sort of, lower animal or other models. And also in the recent years, it became clear that metabolism, and there's going to be also a platform about that, is very different, between different cells. And mostly the model which we used, the human-based model, those were the fibroblasts. But those cells are continuously dividing. They don't have the similarity qua metabolism to tissues, which basically finish developing, very early in an individual, and after that they become, we call them post-mitotic, which means that they never actually divide again, like the heart cells, the brain cells, and maybe even some of the muscle cells. So this makes it very important to pick models, which mimic, or mirror much better the human situation. And the recent advances in the field made it possible to generate human-based models. And how we do it, we take skin cells, and these skin cells can be, we call them, reprogrammed. And at the end, we can have or differentiate brain cells, heart cells, endocrine cells, so like cells which produce hormones. And as you know, these tissues are all severely affected in mitochondrial disorders. And with these new models, we can create a human relevant disease model, and not just human relevant, but also tissue specific models, and study those. Of course, they have their disadvantages because they are just the in vitro models, 2 or 3D models. So they don't have the complexity, but they have the human genotype, because these models have been or actually generated by the human cells. And we think that this could really help us understand the disease mechanism, and also maybe find treatments, because we actually treat cells which are relevant for the disease and also have a human genetic background. And here we put together a panel of experts, and they really have been pioneers and also champions in these newly developed disease models. And then the first one will be by Carla Köhler, and from University of California in Los Angeles. And she will also talk about, the mitochondrial double-stranded RNA and some of these, we call them damage-associated molecules, which get free from the mitochondria, and how that drives the immune response. This has also been a very exciting field of research. And now we have more and more understanding that mitochondria, or especially dysfunctional mitochondria, can indeed drive the human immune system, especially the immune system, which is basically a very evolutionary old kind of immune response. And this, I think, is a very exciting field to understand and also research for the future. Then we move to another talk by Alessandro Prigione, and he is really a pioneer using these human cells, or human skin cell-based models. And he developed brain organoids, so we call them mini brains. These are, small brain tissue, and in this case, he's focusing on the Leigh syndrome, and especially how we can use these model systems to find novel treatments and really in drug discovery. So I think this is going to be a very exciting. I know that he already has some potential drug targets, so we really look forward to his talk. The next talk is also dealing with these cortical organoids. And this is in MELAS, or the most common, pathogenic variant in MELAS disease, and how these cortical organoid systems can help us understand better, the disease itself, and also within these mini brains again. So these are the small brain-like tissue, how different cells or cell types respond, to the mitochondrial bioenergetic defect. And this is also important. I forgot to mention that, of course, fibroblasts, what we used to use for modeling human cells, modeling mitochondrial disorders, are also somewhat different, not just somewhat, very different in the bioenergetics compared to muscle or heart or the brain. So I think this is a very exciting 2 talks about the brain tissue. And then we move to the next speaker. That's Alex Kramerman. He's at Harvard University, and he has developed cardiac cells. And that's another tissue which is, as I said, we call them post-mitotic. So once they develop, they do not divide after that. So basically, the cells that you will have that for your life. And that means also that their metabolism is very different, but also bioenergetically, these cells are really require a lot of energy. You can imagine your heart never stops, beats every minute, 60, 70 times. So this is really energetically a very demanding tissue. And again, understanding how the Melos classical pathological variant affects the cardiac tissue, and then how that can be maybe restored. I believe that these systems will give us better answers and also can be used very nicely for drug screening. So this is going to be a very exciting talk as well. And then we have Graham Preston from Mount Sinai, and he also works with cardiac models, the same variant that the previous speaker will talk about. And this is also Melos disease. And Graham has worked on these for years. And he also used that system, to characterize the cardiac phenotype, but also for drug screening. And he will present his data on drug screening, and then how these systems really helped us, find potential targets, and then which can be moved to the clinic and eventually maybe in a clinical trial. So this is also very exciting. And then we will close the session or the platform by a talk by Quinn Patterson. He's at Mayo Clinic in Rochester. And I think this is really an area which hasn't been really researched a lot. We know that there's a lot of endocrine, so hormonal problems, especially when you talk about, the pancreatic or the tissue, and then diabetes is really a leading phenotype in several of the mitochondrial disorders, but certainly in Melos. And Quinn developed also using human skin cells, developed a endocrine tissue, the pancreatic endocrine tissue, the cell types which really secrete insulin and play a very important role in diabetic phenotypes. So this is again a disease-relevant cell type, human-based cell type, and then we hope we understand better what happens, in these endocrine tissues when you have this classical Melos pathogenic variant. And again, that model could be used then to reverse these phenotypes by potential drugs after using a drug screening. So altogether, I think this platform will give us a very exciting insight, the disease and also cell models, and also the disease relevant cells and phenotype relevant cell types, understanding mitochondrial disorders. So hopefully everybody will join us and for this very exciting platform. Thank you so much.
Hi, I'm Eva Morava, and I also work at the Icahn School of Medicine at Mount Sinai at the Clinical Genetics Division. I would like to introduce you to Platform 2, which is about metabolic plasticity. Metabolic plasticity is an amazing concept because it gives us the option that cells can adapt to their metabolic state with their metabolic status after a certain stress or some kind of altered situation. They are able to activate certain enzymes or change the flux of their pathways and show adaptation to the different environment. This can be used for treatment. This very interesting concept is especially important for our mitochondria. So mitochondria are showing very unique adaptation, not just metabolic adaptation, but they also can change their size, shape, They can move. They can switch to different ways of energy production, and they can undergo fusion and fission by changing their genetic load of a certain genetic background. So with all these different plasticity elements, better understanding this plasticity could help us to find new tools for treatment. Our platform starts with Anu Somolainen, who is a world famous mitochondrial scientist and who has been working on different new therapeutic approaches in mitochondrial disease. She's from the University of Helsinki, Finland. After this, we have a little side trip and visiting cancer. Of course, mitochondria are very important in the development and the treatment of cancer. And cancer cells use their metabolic plasticity to survive and to grow. But understanding the metabolic plasticity in cancer cells can also help us to fight cancer, especially in the phase of proliferation. In cancer cells, mitochondria are very important. Just getting information from mitochondrial metabolism and energy adaptations could help us to use these pathophysiological findings for our own metabolic patients, mitochondrial patients, treatment strategies. The speaker for this talk is Kivanch Birsor from Rockefeller University in New York. The next talk will be about reconfiguring mitochondrial metabolic networks to support cell state transitions. This is really in that topic what I just introduced to you, while different stressors or different drugs could alter the metabolic pathway fluxes, and certain enzymes can moonlight for another and take over functions, which could be advantageous for the energy metabolism. This talk will be given by Lydia Finley, who is from Memorial Sloan Kettering Cancer Center, New York. And the last talk, but not least, is by Ying Fan, who is from Morgridge Institute for Research, University of Wisconsin, Madison. And this will be about mitochondrial metabolism shapes neutrophil fate. This will zoom on some immune cells and their metabolic plasticity, giving us also new potential treatment options. Thank you. Fernando.
Thank you so much, and good morning. I'm Fernando Scalia from the Department of Molecular and Human Genetics at Baylor College of Medicine in Houston. I want to convey the excitement of platform session 3. And platform session 3 will focus on disorders of mitochondrial DNA integrity. And as such, they involve disorders where mitochondrial DNA can be rearranged, such as in the single mitochondrial DNA deletion syndromes, or there could be mitochondrial DNA replication disorders. All of them affect mitochondrial DNA integrity, and these last disorders can lead to multiple mitochondrial DNA deletions and also a decrease in the mitochondrial DNA content that is called mitochondrial DNA depletion. As we know, we may not know, and we still don't know the mechanism of formation of these deletions, and the current treatment options for to maintain mitochondrial DNA maintenance defects are rather limited. So this session will try to address these gaps in knowledge, going from like very different angles. So the first talk actually will focus on mitochondrial DNA rearrangements, mechanisms of formation, and approaches to therapy. And we know that, for example, single mitochondrial DNA deletions are thought to occur as a result of replication errors or following double-strand breaks. However, the exact mechanisms behind the formation of these deletions, single deletions, have yet to be determined. And actually, Dr. Moraes from the University of Miami in Florida, who's a very well-known scientist in mitochondrial disease, has proposed that mitochondrial DNA deletions formed from replication errors versus the ones following double-strand breaks can be mediated by separate pathways. And knowing the mechanism of the formation of these deletions can lead eventually to the discovery of novel therapeutic approaches. The second talk, which is also very exciting, will be a talk given by Dr. Niels Goran Larsson from the Karolinska Institute in Stockholm, Sweden. And actually, it will focus on reprogramming metabolism by inhibition of mitochondrial DNA gene expression. So initially, he had been working on the inhibition of mitochondrial transcription, because there was an observation that the connection between metabolic effects and anti-cancer activity of drugs, of compounds targeting the mitochondria, prompted him and his group to investigate whether the inhibitors of mitochondrial transcription that have documented anti-tumor effects in mouse would also have beneficial metabolic effects. And actually, the past research has allowed him to delineate how this treatment in obese mice with a high-fat diet shifted this metabolism towards fatty acid oxidation, leading to rapidly normalized body weight, markedly improved insulin resistance, and reverse accumulation of fat in the treatment. So this rewiring of metabolism is caused in the liver and provides a novel principle for drug treatment of obesity and obesity-related pathology. The third talk will be a more clinical talk given by a known physician scientist from Children's Hospital of Philadelphia, Dr. Rebecca Ganetsky. And actually, she will be talking about expanding the natural history of pediatric single large-scale mitochondrial DNA deletion syndromes that actually would involve Pearson syndrome or Kearns-Sayre syndrome, and the spectrum of Kearns-Sayre and CPO, chronic progressive external ophthalmoplegia. And expanding the natural history is very relevant because that eventually will help for clinical trial readiness, and also for developing adequate primary outcome measures or endpoints to monitor the efficacy of clinical trials. So this is a very relevant clinical research talk. And along actually this same line of like clinical research talk, Dr. Mark Tarnopolsky, who's a very well-known researcher in mitochondrial exercise physiology, at McMaster University in Ontario, Canada, will actually look specifically at the chronic progressive external ophthalmoplegia spectrum disorder. I think it's very important to see that these conditions may be or may act in a continuum and in a spectrum, and it's very important to classify these patients who would eventually be recruited for clinical trials. Without proper diagnosis, we cannot conduct clinical trials in mitochondrial disease or even establish better primary endpoints and efficacy endpoints. Then following this talk, there will be a study being presented by Elizabeth Reynolds from the CHAMP Foundation in Durham, North Carolina, describing the caregiver burden in families affected by pediatric single large-scale mitochondrial DNA deletions. Her group has published before and published actually a series of surveys to assess caregiving burden. And it's known that caregivers were primarily concerned about what the future held for their children, and the additional burdens included the time required to coordinate the child's healthcare visits and financial strains. So assessing the psychological impact of being diagnosed with mitochondrial disorders is very relevant. Then we shift, in a way, back to the basic science, and which is also connected with research that Dr. Moraes, the first presenter, has done. And this is the idea of like shifting heteroplasmy through finding that in the mitochondrial genome, we can certainly exploit certain weaknesses to do genome editing, because there are sites that actually are restriction sites where enzymes can particularly go, and one particular segment of mitochondrial DNA where a mutation is present. For example, in this case, the common M3243A2G mutation associated with mitochondrial myopathy and with MELAS in the most severe end of the spectrum. Exploiting this weakness would allow us for the first time to do gene editing for mitochondrial disease, expanding the currently available therapeutic approaches, which there aren't many. So the title of the talk is Shifting Heteroplasmy with PB gene, primary mitochondrial myopathy, gene editing therapy for M3243A2G associated mitochondrial myopathy. And this will be presented by Dr. Wendy Shoup from Precision Biosciences from Durham, North Carolina. And finally, the last talk of this session will be given by Maria Falkenberg, Dr. Maria Falkenberg from the University of Gothenburg, Sweden. And she will focus on the molecular consequences of POLG mutations and new avenues for treatment, which there aren't many at the moment. It is actually known that so she has done work on a mouse model for POLG related disorders, and actually she has looked at certain pathogenic variants or mutations in POLG that also strongly impair the interaction with the accessory subunit of the POLG holoenzyme. And when there is a particular, when this interaction is impaired, the free POLG becomes a substrate for a degradation mediated by an enzyme, LONP1. So they are, suggesting, their past research suggests that targeting this turnover of POLG of the holoenzyme by LOMP1 could be exploited as a target for the development of future therapies. Now, I will continue with platform 4. And platform 4 is actually very relevant because, as I mentioned before, we don't have validated or many validated outcome measures for mitochondrial clinical trials. And actually, the first talk, which will be covered by Dr. Shamima Rahman, Dr. Shamima Rahman from the University College of London in the UK, focuses on transforming mitochondrial disease diagnostics through the 100,000 Genomes Program. And I think that this is a very relevant starting point for this session because we have to be able to diagnose or provide a molecular diagnosis to these patients if they are going to be recruited for clinical trials. Previously, Dr. Rahman has actually looked at the value of clinical exome sequencing to diagnose patients with mitochondrial or suspected mitochondrial disease, and the genetic diagnosis was obtained in 1/3 of the cases. And obviously, this could be due to the insufficient coverage for certain regions, and also the sensitivity to detect certain variants using short-read sequencing, or the fact that there could be variants in promoter, intronic, or untranslated regions, and definitely doing a more comprehensive approach through the 100,000 Genomes Project will actually allow to transform the diagnostic approaches for mitochondrial disease. Then the second talk of this particular session will be given by Dr. Tandon from Ultragenics Pharmaceutical in Cambridge, Massachusetts. And actually, Dr. Tandon will review the multi-domain responder index. So Dr. Tandon and the group actually led by him will actually cover this novel approach, and this novel approach is able to accommodate complex and heterogeneous disease manifestations, and it can evaluate a broad array of clinical disease without impairing the power of the study to fully understand the effects of therapy. So really, it combines the results of independent domain endpoints analysis into one responder score. And this actually would allow to provide a broad basis for assessment of efficacy in mitochondrial disorders. And obviously, this would be relevant for other chronic disorders. Then the third talk of this particular session will be covered by Dr. Linda Lowe's from Nationwide Children's Hospital in Columbus, Ohio, and it focuses on the development of a novel outcome measure for rare pediatric neuromuscular diseases. Her research group collects clinical trial readiness data to evaluate current outcome measures and to create novel outcome measures if a suitable measure is not available. And in fact, part of their relevant work was to really develop protocols, outcome measures, selections, and functional data collection. And this actually helped with the approval of gene therapy for spinal muscular atrophy in 2019, because obviously those primary outcome measures were used in that relevant trial. So now, Following that presentation by Dr. Linda Lowe's, we'll have the presentation given by Dr. Zuela Solkipli-Cunningham, who's a well-known physician scientist at CHOP, at Children's Hospital of Philadelphia in Pennsylvania. And the title of the talk will be Validation of Digital Wearables in Mitochondrial Disease. Digital and wearable technologies have been increasingly used in multiple sclerosis and Parkinson's disease clinical trials. But while the assessment of gait analysis has been previously studied in primary mitochondrial disease, the clinical validation against the primary mitochondrial disease specific objective outcome measure has not been studied. And so they have performed assessments using these sensors in patients with genetically confirmed primary mitochondrial disease cohorts compared to healthy controls alongside a validated PMD-specific outcome measure. And this is the MMM COAST, or mitochondrial myopathy composite assessment tool. So this should be actually all of these talks are very exciting and very relevant for the development of improved outcome measures. And finally, the last talk of this session will be given by Dr. Damiano Zanotto from the Stevens Institute of Technology in Hoboken, New Jersey. Actually, Dr. Zanotto has worked on lower extremity, or the development of lower extremity power orthosis for locomotor assistance and rehabilitation, and also the development of wearable sensors and machine learning models for real-world gait monitoring, quantitative assessment of movement disorders, and injury prevention. And the title will be Improving the Assessment of Walking Function in Controlled and Natural Environments with AI-Enabled Insoles. Having covered this, I will now lead to Dr. Yeski, Phil Yeski. Thank you.
Thank you, Fernando, and thank you, Thomas and Ava as well. You know, those summaries you just heard are over 2 days. That's a lot of science in 2 days, and I think it really speaks to the way we try to build out the UMDF conference scientific program to ensure that the people who are coming are really seeing a lot of different science, whether it's basic, translational, clinical, all with a thread towards developing therapeutics for mitochondrial disease patients. And that really brings us to Friday, which is a little bit different. Next slide, please. So on Friday, we try to bring the entire conference together. And so that's researchers, clinicians, patients, families, and industry all in one room to really focus on clinical trials and how we move all of this research towards the development of treatments and cures for mitochondrial disease patients. So we like to start the day on Friday with a focus on the impact of UMDF and what our mission is delivering to the community. Our president and CEO, Brian Harmon, will deliver a State of the Union, sort of where we are and where we're looking to get to, set the roadmap, if you will, for developing these treatments and cures for mitochondrial disease. But inside that session, we'll be calling out some of the highlights of the various aspects of UMDF mission that extend beyond the research mission, whether that's education, support, advocacy. We like to blend it all in there to share with the community the breadth and the depth of the impact of that UMDF is trying to make in the mitochondrial disease community. We also take an opportunity to present some awards, various advocacy and volunteer awards during this time, recognizing the fact that we can't do it all alone. We very much depend on scientific advisors, some of whom are on this call today, as well as many from the patient community themselves and their caregivers that play a big role in helping to deliver the mission of UMDF. So that'll be the first half of the morning on Friday. After we come back from a coffee break, we'll really transition to sort of an annual tradition now at the UMDF conference, and that's a session focused on clinical trials and getting updates around them. So as per usual, we'll have some brief updates from sponsors, investigators that are currently active with clinical trials in mitochondrial disease. But we'll also, or hope, to have a couple of presentations from companies, sponsors that are just coming into the community. And so sort of foreshadowing, if you will, some of the clinical activity that will take place later in 2024 into 25 and beyond. So that's really exciting because that speaks to the kind of pipeline that we're trying to build in mitochondrial disease. We're going to need many different therapeutics to be able to treat the broad range of mitochondrial disease patients. So we need as many sponsors, investigators as possible to be active in developing therapeutics and conducting clinical trials. We may, if we have time, have some brief updates from our clinical research pavilion that I mentioned in the introduction to today's webinar. We see that as a really important way to augment the collection of research data from clinical trials, taking advantage of the fact that we hope to have 200, 250 patient family members at the conference, trying to get them to participate in clinical research, learn about it, overcome any concerns they may have, and then perhaps consider participating in a clinical trial. So I see my colleague Nicole has shared in chat a link to the conference and the clinical research pavilion if you'd like to learn more about that. Then we'll wrap up the session with a number of talks around clinical trial readiness. So if we're building a culture of clinical trials, it takes a sustained long-term commitment to clinical trial readiness. So what does that mean? That means whether you're a clinician taking care of mitochondrial disease patients, researchers that are working on the science, the underlying science to support these clinical trials, patients themselves, we all have to think about what can we do to better understand the therapeutic development process and think about the role we can play in terms of making them more effective. All of this is nudging our community, our rare disease community, towards being better prepared then, being more ready for therapeutic development as outside companies, academic institutions develop promising directions, early stage opportunities to develop therapeutics. So we'll start talking about an initiative that UMDF is stewarding called Treat Mito. That's going to serve as our sort of central hub, if you will, for therapeutic development. That'll be the place where all stakeholders can come together to develop shared priorities that are patient-centric. Here's the place where all of our clinical investigators can come together to consider the opportunities that are presented to our community, help prioritize them, and then identify the best sites and investigators to move those forward and work closely with the patient community to ensure that the outcome measures that are being used are relevant and important to the patient community. This is really an important new initiative. It's our effort to ensure that our research landscape is working in a more integrated way as opposed to in a more siloed way. I think we all know that regardless of what you're working on, better outcomes whenever we work together and we're ensuring that there's good communication across the various ecosystem that it takes to develop treatments and cures. So we'll hear about Treat Mito. We'd also like to share a few vignettes around how our patient registry is being used to advance clinical trial readiness. I think those will be interesting for the community to hear about how we're recruiting patients, helping to curate their genetic testing reports to ensure that we have high fidelity data inside our database about the genotype associated with any patient participant, how we're tying together genotypic and phenotypic information around an important group of mitochondrial disease patients like myopathy patients. And then we'd like to talk about some of the results as well from our no-cost genetic testing programs. And I think everyone knows that over the last several years, we've run a couple of pilots, some patient-facing, some clinician-facing, We now are pulling that data together, and we'd like to share back with the community to show the impact of being able to facilitate the diagnosis of mitochondrial disease patients. This is incredibly important for us to develop as many well-characterized patients as possible to help recruit them and populate the clinical trials that will be advancing both now and in the future. Sure to be an action-packed, very full program on Friday morning. We'll take a little break, come back Friday afternoon, but I think we're going to wrap up this by talking about our Saturday program, if I'm not mistaken. Oh, no, we have Friday afternoon. My apologies. Thomas, you're coming back to talk about that.
So I'm back, and this is really a topic that has been very, try to put on the camera. So this has been a topic which was really fascinated me for a long, long time. And we started to also work on this mitochondrial involvement in and how mitochondrial dysfunction or energy deficiency could impact mental health. And I was very happy that this year's conference, we could have an excellent platform on this. And we will start. And I think it has not been per se recognized, you know, in the clinical care that much, because I think the physical symptoms, they really dominate, you know, the clinical care. And when we talk to patients and also, you know, families, mitochondrial disorders associated with mental health issues, that has really been a big problem. But we still have, you know, limited understanding of the causality, so how mitochondrial dysfunction or energy problems, you know, in the brain, for example, contribute to this mental health issue. But I think there is some excellent research, and this platform will highlight some of those. We will start with the summary of the clinical presentation or the clinical aspect of mitochondrial disease and psychiatric presentations by Amy Goldstein from CHOP at Philadelphia. She has been an expert, you know, in mitochondrial disease and have seen, you know, many patients. And I think that she also perfectly understands and also will talk about the psychiatric aspects of the disease. After that, we will move to another excellent speaker, Dr. Andreazza Anna. She's at University of Toronto. And I know her for many years, more than 10 years, I think. And she has been really a pioneer in the mitochondrial dysfunction, mitochondrial disease, and its impact on mental health and understanding depression, for example, bipolar disorder. So we really look forward to her talk when highlighting, you know, some of the mechanisms and also some causality between mitochondrial function and mental illness. The next speaker will be Mark Fry from Mayo Clinic. He has also been really fascinated by the mitochondrial function. He's a psychiatrist, and he has been fascinated by the mitochondrial dysfunction and mitochondria in general, and what kind of role mitochondria could play in psychiatric conditions. In his talk, I know this story. He actually found a very interesting association between mitochondrial energetics and also how that can elevate the risk for treatment emergent mania. This is in bipolar disorders. And in bipolar disorders, when you treat the patients in depression, in the depressive mood state, you treat them with certain antidepressants. Then they actually suddenly go into an antidepressant-induced mania. And then he found a very strong correlation between a certain type of antidepressants and this treatment emergent mania. And interestingly, those antidepressants which were responsible for this treatment emergent mania, they also were antidepressants which improved mitochondria or actually boosted mitochondrial energetics. So he thought, you know, that antidepressants boosting mitochondrial energetics could result in this treatment emergent mania. Very interesting study, and it also highlights, you know, some individualized medicine or individualized treatment strategies in psychiatric conditions, depending on the patient's bioenergetic status. And then we will close this platform by a talk by Tadafumi Kato. He's from Japan, And he was, I think, one of the really the first scientists who, in animal studies, really established a causality between mitochondrial energetics and bipolar disorders. And since then he published several very high impact papers on the mitochondrial involvement in neurobiological mechanisms of bipolar disorder, but more in general in psychiatric conditions. And I think what we really look forward to this, the end of this session, it's going to be a panel discussion with all the speakers, and we really look forward to the input by the families, by the individuals affected by mitochondrial disorders, and how they live with this condition, and also the psychiatric aspects of it. And hopefully we will also learn what is the unmet need. And I hope that you will also learn from us what could be, the mechanism and how mitochondrial energetics could result in psychiatric conditions. So we look forward to your participation in this platform 6 Friday afternoon.
Thank you, Thomas. And so now that brings us to Saturday. We have one morning session left. And this year we wanted to try to use that to paint a picture of the landscape of where we're at right now on therapeutic development for mitochondrial disease. And importantly, learn from other disease communities about how they've moved rare disease drug development forward. So I'm going to be joined by Dr. Zuela Zokipli-Cunningham from JOP. Hello, Zuela. And we're going to tell you a little bit about this platform and some of the speakers involved in therapeutic development for mitochondrial disease, lessons learned from other rare diseases. So next slide, please. So I'll just start the session by providing that landscape of therapeutic development so far for mitochondrial disease. We won't spend too much time there, but I think it's important for us to know where we've been, where we are now, and then importantly, allow that to help inform where we're trying to get to in having approved therapies. So Zuela, maybe you can talk about some of the other speakers in this session.
Yeah, thank you, Phil. Also worth saying, I think I'm co-chairing this with Fernando on Saturday. So the theme is exactly as Phil said. We want to learn from other rare disorders on what they have done in their journey getting to drug approval. So we're going to be starting the morning with Dr. Richard Finkel speaking. He will be the keynote speaker of the morning. So Dr. Finkel is a child neurologist. He used to be at Children's Hospital Philadelphia, subsequently then in Moores, and now he's faculty at St. Jude's. So he's the director and endowed chair in the Center for Experimental Neurotherapeutics in St. Jude's. The reason he's coming to speak to us is because he's a world expert on spinal muscular atrophy. So another very different but related genetic neuromuscular disorder with generally poor prognosis. So Richard is going to be sharing with us his experience using a very systematic approach in a consortium called the Pediatric Neuromuscular Research Network on SMA studies, which is where I've known him from. And so the systematic approach that the PNCR have applied to clinical trial readiness. 10 years before the first clinical trials were available in SMA, they were prepared with clinical outcome measures, with natural history data, published natural history data, all in the 10 years prior to the first drug being approved in 2016. There's now 3 approved drugs in SMA and a handful are rising to submission to approval. So the world of SMA has really worked hard and made it happen in terms of getting drug approvals. And this is going to be essential learning from us to hear. Dr. Finkel is really highly published on SMA, and he was going to be sharing his experiences with us. The next speaker, who many of us know well, is Dr. Hilary Vernon. So Dr. Vernon is a clinical biochemical geneticist and medical geneticist. She is an associate professor of pediatrics at Hopkins and also an attending physician at Kennedy Krieger, where she sees both mitochondrial patients and Barth syndrome patients. So Hilary Vernon has really an incredible story of how she identified the compound now called elamitritide in her lab, planning the clinical trial, so that entire journey, learning throughout the whole process, using the natural history data that she collected, and she'll share this with us very humbly. It started off collection of data on a spreadsheet, not quite the databases and mechanisms we have now. and then how she conducted the clinical trial, getting to good results, and more recently, getting to submission of the NDA to the FDA. And so they are working hard to get drug approval in Barr syndrome. It's a very closely related disorder to mitochondrial disease, and this will be a terrific talk that she will be sharing with us. The next speaker that we have invited is Dr. David Lynch. Dr. Lynch is an adult neurologist who is working at Children's Hospital of Philadelphia. He is a world leader in Friedreich's ataxia, both in clinical diagnosis and clinical management, as well as clinical research activities. He follows a very large Friedreich's ataxia cohort, about 300 patients. He's been pushing forward and very active in designing and making sure clinical trials are conducted to a very rigorous standard. And he's also active in bench work. He's actually a bench work researcher. So collectively, all of his research efforts have been very, very strong in Friedreich's ataxia. And most recently, FA now have their first drug approval, Skyclarys. So there is a lot to learn from the FA community on how they advocate very strongly for their patients to enroll and participate in clinical trials. Dave shares with me their tremendous effort at educating their patients and families on the importance of participating. So we will be hearing these experiences from Dr. Lynch on the day. The last and final speaker of the morning before the roundtable discussion is Dr. David Whiteman. We are delighted to invite Dr. Whiteman to speak at our this session. He is a medical geneticist. 25 years ago, he states, he ran the metabolism program at Mayo. And he was, in fact, also an active clinician in the mito disease community and was on the UMDF Scientific Advisory Board many years ago. His last role was at Takeda Pharmaceuticals. He has extensive experience over decades on rare disorders drug development. Back then, mitochondrial drug development was not as active. So Dr. Whiteman's coming to speak to us on lessons learned from other rare disease drug development programs and thoughts. He's going to be sharing his thoughts on how to go about clinical development for some of the emerging mitochondrial therapies. So he will be the last platform speaker of the morning. We are then going to have a panel discussion. And Phil, I don't know if you want to jump in here.
Sure.
The panel discussion, Phil was really eager to have representatives from foundations and pharma. Sorry, Phil.
No, I appreciate you mentioning that. Yes, we're really thrilled that we're going to have representatives from Barr Syndrome Foundation and Friedreich's Ataxia Research Alliance, two patient advocacy groups that we very much respect and appreciate their collaborative nature and how they work together with UMDF to try to advance the care and therapeutic development for mitochondrial disease patients. So Lindsay Marjoram and Ruth Acton will be joining from those organizations respectively. Then we'll also have participation from industry, from Biogen, where they've developed a therapeutic and have a marketed therapeutic for Friedreich's ataxia, Angela Paradis. And I think these additional speakers, in addition to those from the platform, will really provide a complete perspective on the development of therapeutics for rare disease. This is a tremendous opportunity for us to learn from other disease communities and really try to position mitochondrial disease for the same kind of success going forward. So thank you, Zuela, for your help in putting this together. And I think that'll be a great way to wrap up the conference on Saturday morning. So with that, I see we are just about at the top of hour. I'd like to thank very much Thomas, Ava, Fernando, Zuela for all your help in putting this together. There are a couple other clinicians that contributed significantly to putting the program together this year. Sumit Parikh was not able to join us, nor was Jody Nunari. So thank you to them as well for identifying speakers, developing the platforms. Really excited about the program that we have put together for this year. And we haven't even talked about the family program. There's many exciting opportunities on that side too for those who are attending the conference. So we look forward to seeing as many of you as possible in Cleveland, June 26th to 29th. So with that, I'm going to bring the webinar to a close and remind you that we take a break with conference, summer holiday. We will not have Bench to Bedside in June, July, or August, but we'll be back in September of 2024 with more great programming, and we look forward to seeing many of you then. So thank you again.